Hello, and welcome to our panel discussion here at the ARM meeting on the Mesa, meeting virtually rather than in person in Carlsbad, California. And I would like to introduce our topic, which is current hot topics and emerging trends in gene therapy. And our panelists are Kathy High, Natalie Hollis, and Ken Mills. And Kathy, I'll turn it over to you to introduce yourself, and then Natalie, and then Ken.
Thanks, Bob, and greetings to the virtual audience. My name is Kathy High. I've been involved in the field of gene therapy for a number of decades now. I started my career on university faculties and was on the faculty at the University of Pennsylvania and the staff at the Children's Hospital of Philadelphia until 2014. I headed up a center at the Children's Hospital of Philadelphia that was devoted to establishing experimental bases for cell and gene therapies, particularly for inherited diseases. And in 2013, we formed a company based on a number of the studies that we had been conducting at the Children's Hospital of Philadelphia. And that company, Spark Therapeutics, was formed in March of 2013, and in September of 2014, I left my position as a Howard Hughes investigator to become the president and head of R&D at Spark Therapeutics. We were fortunate in our work to actually developed the first approved AAV gene therapy in the United States, Luxturna, for a rare form of congenital blindness. And we achieved breakthrough therapy designation for 2 other programs in hemophilia B and hemophilia A. In December of 2019, Spark was acquired by Roche. And in February of 2020, I retired from the company and am now acting as a consultant for a number of different groups.
Well, that's a tough act to follow. Thanks, Kathy. Natalie Hollis, I'm the president and CEO of Audentes Therapeutics. I have been with the company since its early days, about 5 years now. I initially joined as the chief operating officer, and one of my early and very exciting tasks was building out and commissioning our large-scale GMP AAV manufacturing facility, which is still one of our proudest achievements as a company. Like Spark, we were acquired. Actually, our deal was announced in December of 2019 and closed in January of this year. We were acquired by Astellas and have become their gene therapy center of excellence. And it is a fantastic relationship that has been really accretive to the overall value of what we're trying to do at Audentas. It's been a tough year for us, a very difficult year in our lead program in X-linked myotubular myopathy. As most of the ARM community is likely aware, we lost 3 little boys in our clinical trial this year and are currently on clinical hold while we do the work to understand what happened and chart a path forward for what we still believe to be a really transformative medicine, but to chart a path forward that it ensures the safety of all the boys that we want to treat eventually. Besides the XLMTM program, we're very excited to have our program in Pompe disease heading into the clinic shortly. And we've got late-stage preclinical programs ongoing in Duchenne muscular dystrophy and myotonic dystrophy type 1. And we have big dreams to expand the technology and follow AAV wherever the science takes us. And to do that as part of a big global organization that is fiercely supportive of the mission is really a fantastic place to be. So I'm happy to be here to join the discussion today.
Great. Hi, I'm Ken Mills, the President and CEO of REGENXBIO. Really pleased to participate with Kathy and Natalie and Bob in this panel today. REGENXBIO was formed in 2009 with a focus on delivering on the curative potential of gene therapy. Been involved with the company since the beginning as one of the founders, and we've had several chapters in the history of the company, which has included everything from aggregation of important intellectual property to licensing and partnership. And today the company has over 300 employees, mostly based here in Rockville, Maryland, working on a pipeline of therapies that are clinical stage across 3 therapeutic areas: retinal disease, neurodegenerative diseases, and metabolic diseases. As I mentioned, the history of the company has had components of it that's involved a lot of partnership, and we also have a pipeline of partnered programs, including through and in cooperation with some of my colleagues on the panel today, that include programs that are now part of large pharma pipelines, as well as SMID-cap pipelines, all focused on advancing AAV gene therapy towards that mission of new treatments for patients.
Well, thank you, Ken. And Natalie and Kathy, I'm Bob Smith, Senior Vice President of the Global Gene Therapy business at Pfizer, which operates within our rare disease business unit. We've been investing in gene therapy since 2014, and in fact, our first significant investment was a collaboration and license agreement with Kathy and the Spark team that we entered into in December of 2014, which has now led to our ongoing phase 3 program in hemophilia B. We also have another hemophilia program that's in phase 3 for hemophilia A in collaboration with Sangamo Therapeutics, and we'll be soon starting a phase 3 trial in Duchenne's muscular dystrophy. And similar to the investments that Natalie and Audentes and now Astellas have made in gene therapy manufacturing, we have 3 dedicated gene therapy manufacturing facilities with a total capital expenditure investment of more than $800 million, where we're producing across the spectrum of research grade, research scale through early clinical GMP scale. And then now we're building out a dedicated commercial facility that'll be online next year. And, you know, similar to the experiences that I think all 4 of us have had, I describe it as every day is an adventure in gene therapy. And even as early as just a few hours ago, you know, another gene therapy company went public, Taysha, and we've seen a significant explosion in the activity, not just in terms of scientific advancements, clinical development milestones, significant capital investments in manufacturing, regulatory achievements, and the investor appetite for gene therapy, particularly in the COVID pandemic, has been quite remarkable. And we'll touch on all of these topics during our panel. And maybe the first topic will be, if you could each share some of your insights into some of the notable scientific breakthroughs, some of the challenges that we still have remaining in the field of gene therapy, and you know, have a discussion about that. Then we'll move on to the clinical aspects. So Kathy, maybe if you want to get us started on that particular topic.
Well, when I think about scientific breakthroughs around gene therapy, one of the things that I really feel good about is that we continue to see momentum in terms of both approval of new products, which you may not think of as science, but I think of it as science, but both in terms of approval of new products and also reports at scientific and medical conferences about results in clinical trials that, you know, seem very compelling. My background is actually as an internist and a hematologist. So I have to say one of the reports that I've found most exciting is the data around new treatments for transfusion-dependent thalassemias and for sickle cell disease. And, you know, I think these 2 diseases illustrate some of the power of gene therapy. We did have a treatment for transfusion-dependent thalassemia, which is transfusion and then iron chelation. But that's an example of a treatment that is so complicated and so difficult that the burden of the treatment itself presents a pretty significant obstacle for the patient and the family to compliance. And so if you think about what gene therapy can do, for example, Bluebird's program in transfusion-dependent thalassemia, now the patient can undergo a fairly intensive medical intervention for a short period of time, bone marrow transplant with gene-modified autologous cells, and then reach a new level of health where they don't need nearly as much interaction with the medical system or medical intervention. And I think, that's really what we're looking for. I think that the work that's gone on both for Bluebird and for CRISPR, which uses a different approach, a genome editing approach rather than a lentiviral gene delivery approach for sickle cell disease. Now we're talking about a disease where we really haven't had disease modifying treatments of any kind. So now you're talking about gene therapy creating a treatment for a disease that we really didn't have disease modifying treatments for. And so I find that very scientifically exciting. I think in terms of other scientific developments over the last year or a couple of years, we're seeing more and more work on a novel technology of base editing. So rather than genome editing as with CRISPR, now this is actually using a technique to correct a single base. And this, although this is early and we don't have clinical data with it yet, it provides another avenue for approaches to genetic disease and probably eventually to complex acquired disorders as well, genetic strategies for complex acquired disorders. So I think it's been a busy time. It's been a good time.
Yeah, it's certainly remarkable how quickly some of these technologies are emerging as well, and the pace that things are moving from essentially taking a technology tool, going through a very accelerated translational preclinical research paradigm and potentially quickly into the clinic. I continue to be astounded just compared to the years I spent working with small molecules and biologics, that the time periods are becoming more and more accelerated. It's really quite remarkable. Ken, any other areas where you find scientific breakthrough being exciting for yourself and your organization?
Yeah, I think I would stand on top of two overlapping points that Kathy made and that you made, Bob, which is, and medicine is absolutely science. And I think that, the development of approved products and the number of new product candidates and the rate at which new product candidates are coming forward is really unbelievable. And almost in a scatter plot form, right? I think the number of new tissue targets, the number of new modalities, Kathy referring to things like gene editing, individual base editing, but also, just gene knockdown with other modalities that have been around for a while, maybe with different types of routes of administration, are all sort of overlapping on this foundational, new and safe platform. And I think what's really cool to see with the science is, and I think what's unique about gene therapy, whether you're talking about attributes of AAV gene therapy or the ex vivo approaches, is the reuse, right? And talking to your point, Bob, about, things in the fields where small molecules or biologics were the sort of tools that clinical scientists and medicinal scientists had, they had to sort of stay in these narrow bands, test and retest all the same things for each one of those new molecules. But here there's so much building upon the de-risking that's gone on for decades, as Kathy's alluded to in her own introduction, and now on a very accelerated basis in the last 5 to 10 years. So that effect is having really geometric expansion in the number of patients that are able to get treatments in now clinical trials and the emergence of these new amazing approved products.
Yeah, this is all great points, Ken. Natalie, anything from your perspective?
Well, I'm going to start with a story. Actually, I'm going to build off something you said in your introduction, Bob, which was that you talked with Kathy about licensing her hemophilia program back in 2014. And Kathy, you won't remember this, but I was with another rare disease company at the time called Hyperion Therapeutics. And we actually had a call with you to discuss your hemophilia program. And the data were beautiful. But my scientists and clinicians in 2014 were like, oh no, gene therapy, I mean, it'll never happen. It's just way too risky. Let's go find something else to do. And so we passed, And here we are 6 years later. And as Bob was saying, you know, there's a cellular gene therapy company going public approximately every 4 seconds. And there's just an explosion in terms of the number of clinical and preclinical programs going on. And so if you just think at the highest level about the pace of growth and acceleration in gene therapy, just in my short tenure in the field, just 5 years, it's really astounding and really gratifying. When I thought about taking this job in 2015, an old mentor of mine said, "Oh yeah, gene therapy, medicine of the future, and always will be." And the fact that here we are and we actually have approved products, it's pretty incredible. So I think that's a testament to how quickly the field has expanded. You know, what I'll say in terms of the technological advances is, you know, I grew up on the business side of the industry. And so I always think about things in terms of how can we get the broadest access to the most patients with any particular modality. And 2 of the limiting factors in gene therapy to date have been pre-existing immunity and the ability to retreat. They are 2 of the tough concepts that we think about and talk about a lot internally and always have. And so I think some of the interesting technologies that are evolving around capsid engineering, immune suppression regimens and things like that, that remove those kind of structural barriers to being able to treat broadly, particularly in adult populations where pre-existing AAV immunity is more prevalent, I think is really exciting. We're looking at it in the context of our Pompe program. And I think that that's going to be a really interesting direction of evolution in the field, because even with our existing tools and even the monogenic diseases that we're trying to treat, if we can make it so that we can treat every patient who has the biologic defect, I think that will be a huge advancement for the field.
Yeah, fully agree. And it's kind of interesting, and Kathy, maybe we can just reflect on our collaboration on the hemophilia B program, which is the early design of the clinical studies and the inclusion/exclusion criteria and the clinical protocol. We're really designed with achieving as safe a product profile as we could in a patient population with the highest feasibility of having a clinical benefit. And, you know, as a consequence of that, you end up with an inability to treat all potential patients. So, you know, we fully agree with your comments, Natalie, that coming up with different technological improvements to the limitations, particularly with AAV in vivo delivery, would be a significant advancement, because there is going to be some proportion of patients in almost every disease where we find this to be a tractable therapy that won't be treated under the current limitations. And that I think is something that the field really needs to amp up its level of investment. I know that you guys are working on it. We're also working on it. And you know, hopefully we'll see some pretty significant discoveries there that will help address some of these limitations.
Yeah, I would add, Bob, that, you know, I have those conversations a lot with a number of stakeholders in the field, and I often find that, you know, some of that is still rooted in what route of administration we're talking about, and certainly, as you know, Natalie alluded to, the age of the population. You know, there are clearly no treatments, you know, in any modality that really can get to all patients. It's great aspirationally, and I think a lot of these markets, and we look at, you know, what's going on in our field in hemophilia A or in muscular dystrophy or Pompe. I mean, there are a lot of candidates coming forward. I think that's a good thing. That's a great thing for patients, not just to have competitive options per se, but also to have options that of availability for them on a going forward basis. But we also have experiences in our pipeline at REGENXBIO with certain routes of administration. For instance, our wet age-related macular degeneration program with the subretinal route of administration, where we don't screen for things like neutralizing antibodies. And when we have prospectively or retrospectively done so, it has not had any sort of effect in that compartment where that delivery is occurring for that disease. So, you know, it's a variation on a theme, incredibly important to be, you know, focusing on these issues, especially in certain disease areas, because again, I agree, this foundation is so great to build off of, but you know, let's not rest on our laurels here. Let's keep improving.
Yeah.
Yeah, and I would just add to that point, from my perspective as a hematologist, one thing I can tell you is you can always get rid of antibodies. There are lots of different ways to do it. You don't need to get rid of them for very long, right? You just need to get rid of them for a little while so that you can infuse the vector. So I would be hopeful that we will be able to devise a solution that at least takes care of the antibody problem. As you know, the immune system has many components, and so that may not be the only issue, but I think that will be a hurdle that can be overcome. All of us on this panel have a lot of experience with AAV, but I'm just gonna say, I think there have been other interesting scientific developments over the last couple of years that aren't AAV centric. And one of those that I would name is that, sort of as a follow on to the very exciting work in CAR Ts, which are mostly autologous cells, that we're beginning to see data coming out with sort of universal off-the-shelf T cell therapies. And that I think would really expand the utility of these kinds of approaches. So I'm glad to see that work going forward with multiple different sponsors. And so I think that to pick up on Ken's point about more people volunteering and then more people eligible, I think if we have a situation where we don't have to actually extract the patient cells and manipulate those in the lab and return them, but rather have an off-the-shelf approach, that would be a real advance.
Yeah, totally agree, Kathy. I'm just kind of curious also, Kathy, if you had thoughts on non-viral vector-mediated ways to deliver gene therapies. If that's something that is potentially on the horizon, do you see any exciting leads there that may transform the field even further?
I mean, certainly we know that there have been a number of IPOs that have gone forward with non-viral gene delivery. So there are certainly people very actively engaged in the space. I would say that having spent decades of my career wrestling with the human immune response to AAV, no one would be more enthusiastic about efficient non-viral gene delivery than I would. But I do think it's also difficult in some ways to match the efficiency of a virus that's had millions and millions of years to evolve to reach the goal of efficient delivery into a eukaryotic cell. And so, I mean, I'm very hopeful that this will be the direction that the field moves. And then all of these issues that we're talking about antibodies or T-cell response, will just go away. But you know, it's been a slow road so far.
Yeah. Any remaining thoughts on exciting opportunities or challenges on the scientific technical front before we move on to clinical?
I'm going to defer to Kathy on that. I think she's the guru in this category.
I think, well, there are lots of exciting things going on, but I think we've covered a number of them, Bob. I think we're.
Well, why don't we move on to the clinical segment of our panel? Ken, do you want to make a few opening remarks for us?
Yeah, well, maybe, Bob, we can bridge from science to clinic by way of, route of administration and tissue targeting. I do think that that's an area of science that has, dramatically expanded, in part based on, better understanding and better availability and resources for how things like AAVs, but even, some of the other technologies that Kathy has referred to, I think some phenomenal, first-time things are occurring in gene therapy with, targeting new tissue types with new surgical interventions and, borrowing the biology and the sort of, concepts of tropism around the, viruses themselves, but really working to safely get things closer to the tissue target. And we see that in, especially, I think, neurology and neurodegenerative conditions. I think we see it throughout the CNS. I think there's even cases of it in ocular indications where, people are continuing to try to improve the, delivery from a route of administration to make things safer and more effective. So that's one area I'd be interested for people to weigh in on, even with their own pipelines and experience. I think another thing that I've observed in the field, particularly over the last 5 years, is a lot of companies, and I think all of you are involved in making that shift from first in human to later stage in development in the clinic. And while some of those trials are really informative at first in human stage, they're getting bigger in later stages. They're requiring more involvement of expertise in clinical operations, more sites, more focus on design overall. And I think that's a phenomena that, while the industry at large has been comfortable with for years, it's new for gene therapy, and gene therapy's got some, new variations on that theme, and regulators do as well. So, those types of transitions. And then the last piece is certainly, where is this science taking us with new diseases? I mean, we are literally doing new clinical studies, in some cases in diseases that, maybe haven't even seen treatments before. And so, between route of administration and moving some studies into, larger phase 3 pivotal trials and new diseases, a lot of exciting things going on in gene therapy. And, I see some of these themes affecting every company, especially those of us that, have been involved in this field for the last 5 years, needing to make transitions with our people and our operations to plan for products.
Yeah, well said, Ken. I was actually reviewing the ARM first half 2020 annual report on the status of the regenerative medicine field, and it's amazing to me that there's almost 1,100 ongoing clinical trials across the regenerative medicine space, and of that, there's 359 in gene therapy, and of those, there's 35 that are now in phase 3. So, to your point, it's really phenomenal growth, and frankly, a relatively short period of time. And I, at least from the experience here at Pfizer, where we've now advanced a number of programs into later stage trials, especially operating in the COVID-19 pandemic arena, it's even magnified the challenges of doing efficient clinical development work in certain parts of the world. And we've taken an approach of operating some of our studies in more than 50 countries, so we have pretty broad geographic diversity, which also then adds another level of complexity when you're trying to operate in the COVID pandemic.
I found, though, one element that in our experience has made clinical operations easier now than it was a few years ago is with the breadth and depth of clinical work going on in the field, you have more clinical sites, more institutions that have gene therapy experience. And so we're finding that the cycles on IRBs and IBCs and just getting over institutional hurdles to start a clinical study in gene therapy, which could take well over a year when we started 4 or 5 years ago. Now that those sort of grooves have been set, and so that's actually making it easier to get our studies up and going more broadly. So I think that that's one benefit from the breadth of the field. I think one of the areas that's interesting for us as we move into Pompe and Duchenne in particular, where there are other therapies available, where these are complex progressive diseases, thinking about study design and endpoint selection in order to be able to show treatment effect, not only in terms of biomarkers or on biopsy data, but actual clinical benefit, kind of being able to tease out the signal from the noise in order to demonstrate the value that's really going to be important to be able to successfully commercialize these products, is an area where we spend a lot of time. There's actually a lot of innovation in that area, I'm finding. But I'd be curious to know, Bob, your experience in Duchenne and even Kathy, as Spark and Roche think about Pompe, if you're finding that those are interesting questions that are evolving as the work gets broader.
Yeah, I can certainly offer some perspective. I think the challenge, not just on individual programs, but potentially in groupings of diseases where there is no effective standard of care, selecting the endpoints, not just for regulatory purposes, but more impactfully for really measuring a transformational or potentially transformational clinical benefit is really important. And it kind of runs the spectrum of things that are kind of standard, almost like cookbook-like clinical development endpoints, to things that could be potentially really innovative with digital monitoring, maybe even on a 24-hour-a-day basis. So we've been looking at innovation across the spectrum of the clinical development studies, from how we do them to how we measure them to how we analyze it to how we package that in terms of putting together a value proposition that would be important for payers as well as the patients themselves. So a lot of work in that regard. And I think as we do each individual program, we can apply the learnings from the first one to the second one to the third one. And so we're kind of like getting on a steeper part of the learning curve, and hopefully that will continue to accelerate.
Yeah. And we're finding a lot of really good input from patients in endpoint selection and design, things that really impact quality of life from the patient perspective, which I think is a great evolution in the way that we think about trial design and endpoint selection.
Yes, yes.
Well, I'll just add in that certainly for Luxturna, which was the first treatment for any inherited retinal dystrophy, we did wrestle with the problem of what would be an acceptable endpoint. And we did, to echo the themes of the other panelists, we did find that the regulators were very interested in not just measures of visual function, but some measure of functional vision. How does this intervention change the patient's ability to execute some visually dependent activity of daily living? Yes. And so we came up with a mobility test that was conducted at different levels of light, which really reflects the defect in the disease, relative lack of light sensitivity. But then we found that not only did we need to develop that, but we needed to conduct a separate study to determine the performance characteristics of the test and to validate the test in terms of reliability and reproducibility and so forth. One of the things we learned in the study of the performance characteristics, because we had to relate it to more conventional methods of measuring visual function, like visual acuity, like visual fields, like light sensitivity, we were able to show a good correlation with light sensitivity. And that was important for us in the reimbursement programs that we designed, because light sensitivity is something that can be measured in the ophthalmologist's office. You don't need a special mobility test. And then we were able to use that in our rebate arrangement program if patients did not get a clinical effect from the treatment, and if the clinical effect wasn't durable, but we knew that this light sensitivity correlated well with our primary endpoint. So that's what we actually used to assess that clinical effect. So, Bob, just to relate back to your point about endpoints that have, around which you can build programs for the payers.
I'll maybe extend from that great Luxturna example and switch lanes to, you know, something outside of rare diseases. And you know, again, just to paint the picture of what it feels like to be taking gene therapy into a well-established market with well-established products and expectations from regulators on endpoints. In our, you know, wet age-related macular degeneration program, you know, it's unequivocal that we're not going to be creating any new endpoints here for patients. Although there are things that we want to measure for gene therapy that are new, and I think, you know, will contribute to some of the value propositions that we continue to appeal to for payers and patients themselves and physicians. But, you know, we walk into a space where, you know, we have the same responsibility to clinical endpoints and primary endpoints like best corrected visual acuity that, you know, the nth generation of re-injectable VEGF inhibitor will have, or, you know, even these device products that are coming forward that elute those. And, you know, it's an interesting place to be because, you know, again, it emphasizes, I think, an important point that we've all talked about implicitly or explicitly about gene therapy, safety, right? I mean, you have to have safety in areas like this in particular. And there are specific examples right now outside of gene therapy where, things like ocular inflammation, with other well-established products, minute degrees different from pre-existing standard of care are having effect on their adoption, support by physician communities. Imagine that, right? I mean, Kathy, I don't know from your experience, I mean, imagine, having to come into that and being able to say that actually we think today that gene therapy is as safe as some of these other things with all of the things that come along with it, but can stand up against other treatments that are literally being delivered to millions of patients worldwide. It's just a very different feeling and flavor. And I'd say it's almost, there's bipolarity some days at REGENXBIO, because I swing back over to our, CNS directed MPS2 program. And, Natalie, to your point about, I know a lot of the diseases you're focused on at Astellas, Audentes, we're working with, physicians, we're working with patients, we're being creative ourselves, much in the way that I think, the pioneering work behind Luxturna went into, not just relying on what's known natural history-wise and trying to shape it into clinically meaningful understanding, but adding new components to that and collecting them, Bob, like you referred to, with new technologies. So, it's pretty cool that gene therapy as a modality can fit in across that spectrum, and, I think no less challenging.
But I would really underscore Natalie's point about that it's becoming a much more accepted modality. I can remember when we were beginning these kinds of studies, I would always have to, not always, but often have to go to the institution, give grand rounds, try to talk to various people who were in decision-making positions around clinical work at the institution, because there was so much trepidation about gene therapy. But, as people gain experience with it, to your point, Ken, they realize it's, it's not something that has a safety profile radically different from other classes of therapeutics, when it's so I think that,
when the effect is so transformative, just the risk benefit calculus is, you're willing to consider things when you can see that. I mean, I still remember those videos of your kids doing the mazes, Kathy. I mean, that was just one of the most incredible things I'd ever seen. Or the Avexis videos from the SMA kids, even our XLMTM videos. I mean, it's incredible. And so you see the power, the potential of the technology combined with a lot of experience now, and it's a much easier road. I don't know if you guys all remember the rack. Do you remember the rack that we had to go through before you could start a clinical trial? That only met 4 times a year, and God help you if you missed it and had to wait another quarter when you were a publicly traded company. That feels like a long time ago, but we lived or died by the rack in the early years. So it's, we've come a long way. We've come a long way.
Yeah, one of the interesting things, even though we've made significant advancements, I'll just share some interactions that we've had where there is still concern because of the irreversible nature of the gene therapy, that safety will always be a parameter that there'll be an uncompromising level of quality on. And I think a lot of the regulatory guidances that have come out in the last few years, not just here in the US with the FDA, but also in other jurisdictions, I think having that very rigorous kind of safety requirements is something that, as we continue to expand the field in the number of diseases, there'll obviously be things that we don't know, the unknowns that you don't know about. And so I think that's where the field really needs to remain vigilant. And, we're hopeful that, to your point, Natalie, the transformational benefits will continue to outweigh the risks.
Yeah. I think transparency and, sharing experience is important as well. I know there's a lot of interest in the field and concern, understandably, with our experience in XLNTM this year. And we've said we're very committed to sharing the data and the experience broadly with the field once we have it all put together. And I think those things are going to be important so that we can learn collectively, particularly when it impacts patient safety.
Yeah. Maybe just turning to another segment of our panel discussion is manufacturing, which we as practitioners in the field know is a significant area of both investment and challenge, and also quite frankly, I think we've seen significant advancements in the ability to scale, the robustness of manufacturing processes, increases in the level of sophistication of different analytical methods that impact and measure product quality and different attributes of products. So maybe we'll just open it up with that point. I know that different companies are pursuing different strategies, whether internal or using external CDMOs as an example, or some blend across that spectrum. So welcome your thoughts as to how you view the expanding and advancing field of gene therapy manufacturing.
Well, maybe I'll just start and say that it's really interesting to me that this has been a bottleneck for decades. In other words, when in the 1990s, when we were first beginning gene therapy trials for hemophilia, we had to scan the whole United States looking for a manufacturer who could make enough recombinant AAV that we could actually begin gene therapy trials in a 70-kilogram person. And that here we are, more than 2 decades later, and every contract manufacturing organization that you can name has a queue, that's long. We always felt at Spark that it was very important to have our own internal manufacturing capabilities, but I can still say that the effort involved in moving our clinical grade manufacturing that we had had for a decade at SHOP to a commercial level was, a very heavy lift. I mean, it was similar in difficulty to getting the product approved. And of course, it meant that FDA inspections, EMA inspections, and so forth. So it was, it was a great deal of effort on the part of a lot of talented people from regulatory and manufacturing and quality and all of those working together to meet that bar.
I'm surprised too. I mean, the number of calls I get from companies, small gene therapy companies saying, I think we need to build our own. Like, how did you do that? How do I do that? And I thought for sure that by now the capacity would have caught up with the demand. But I think that the field is growing. Its rate of growth is exceeding the capacity out there. It's still a bottleneck. And so more and more companies are making the decision to bring it in-house and go it alone. And, I hope over time that we get enough scale that to me feels very inefficient. Everybody's sort of building their own mousetrap. And as you said, Kathy, when you start at small scale, eventually you've got to change your process and scale up. And that introduces regulatory complexity, clinical complexity, or you do what we did and you just start at full scale and just keep your fingers crossed every time and do it under GMP conditions and hope that you get some vector out the other side, which was our way of working in the early days. So I think it's interesting. And then even the fact that there's still really only one shop in the country, in the world, that you get plasmid from. And everybody is bottlenecked waiting to get their plasmids from North Dakota. I'm sort of kicking myself that I didn't open Natalie's House of Plasmids. I would be retired by now if I'd thought of that idea. But it's interesting the degree to which capacity is still a bottleneck. I hope that it fuels innovation in terms of more efficient production systems and just ways to get more vector out of each liter of bioreactor or each Astellas plate or however you're doing it. But I agree with you. I still think it's a bottleneck and we're still behind in that part in terms of keeping up with the demand for vector.
But I do think you make a good point, Natalie, that it's not that the manufacturers have been standing still, right? They've been ramping up and ramping up and ramping up. But
Yeah.
when you see 35 trials in phase 3 and 359 INDs in gene therapy, you know, the level of activity in the space is outstripping the manufacturing capability.
Yeah, they just literally can't keep up.
No.
I think I would say that even in our experience, you know, where our model has been, I think, to be a first mover in, you know, building supply with partners in the CMO network. And it definitely, it does look and feel very different today than it did 5 years ago, you know, 6, 7 years ago, even when we were working with some of our first partners. And we have moved around a lot, you know, across partners. It's always been important, I think, for us to bring our process and work with others to install that. And I think, you know, part of the bottleneck comes from the training of the people and the quality of the people that they're able to apply to your processes, right? And, you know, so even people can be building these rooms to support bioreactors or hyperstacks or, you know, they may get that, but the team needs to be the same. We know, I mean, I say this to our team across every part of the company in gene therapy, continuity is so important. And it's particularly true on the manufacturing side. And, you know, to your point, Natalie, about upstream, you know, things like plasmid and supply chain, you know, there's also the downstream, the final fill and finish, the things that go beyond the bioreactor and the first order purification that also are, you know, and can be unique if you introduce new routes of administration. Like we've got our subretinal delivery, we've got suprachoroidal delivery, we've got our CNS delivery, and we've got IV administration. We need to have a supply chain that can support all of that. And, you know, and we're a 300-person company. So, you know, I feel good, and I'm glad that we were a first mover, because I wouldn't like to play behind here. And I think those that got caught in the middle, who probably should have gone the direction like all of you did, which is to build, recognize that there maybe was a glut in the supply chain already. You know, some people did get stuck. It took them too long to build, or they, you know, they got boxed out because of reservation fee concepts and other things that started to come forward that, you know, some of us had never even heard of before.
They didn't exist. It's opportunistic fees that are getting imposed.
I do, you know, and I worry, you know, I mean, as much as we've been talking about some of the, you know, foundational things that the companies are doing to change that in the supply chain, there's also this, you know, grassroots world in gene therapy as well, still at, maybe more so than ever, Kathy, at the academic translational centers, you know, where individual patient advocacy organizations are looking for a place to make their stuff as well. And that's added a whole nother new competitive dynamic that, you know, wasn't there 10 years ago. And they have money to spend in some cases as well. It's very competitive. I have to remark to the three of you, the companies you've been involved with, you know, I think you've all done things the right way. And the engagement I've had with those of you here, you know, very thoughtful, always putting quality first as well for patients on this.
Yeah, that's important as well.
I'll just share a little story of what I call my experience of asking our Pfizer senior management for the capital investments to build out our gene therapy facilities. And one of the questions we got was, well, show me the clinical data to support this investment. I said, well, we're not in the clinic yet. So we're going to be building essentially a facility to produce pivotal material and commercial material before we had actually put our most volume dependent product into the clinic. And they said, well, what do you call this strategy? And I said, it's called planning for catastrophic success.
Love that.
Which is that if you don't plan your manufacturing and supply chain, assuming that you're going to be successful and have it in place so that you could quickly move from a phase 1 program into a phase 3 program without spending a year or 2 years or maybe longer to build out that larger scale manufacturing capacity, you really do hamstring the program. And I think, you know, to the detriment, not just of the program, but also the patients who could potentially be treated, that assuming you're successful. So that's been our approach. We're still. I
had a similar story about my first board meeting at Audentes, and I had to ask a bunch of venture capitalists to let us invest the CapEx to build out a plant, and we were still a year and a half from the clinic. It was an audacious ask, but they went for it. But it is, I mean, it feels so cart before the horse, but you're absolutely right. You don't want to get into the clinic, have stunning data, and then have to sit for a couple of years while you get your manufacturing to catch up.
But, I do think that connects back to what we were saying earlier. That's one of the unique aspects of gene therapy is that if it's a genetic disease, you supply the gene, you can really have dramatic clinical results in phase 1, 2, and then boom, you need to be ready for phase 3. And that is, I think, I don't think that that's absolutely unique to gene therapy, but I think one sees that quite commonly in gene therapy, that phase 1, 2 results are clear cut, and it's time to design phase 3. So
there you go. And then you've got to play catch up on every other element of the program.
So I think that is really a difference for people accustomed to working in CMC for other classes of therapeutics, is that all of a sudden, wow, I need to have all of this stuff locked down.
Yep.
Let me just pivot to our last segment here, which is on financing and the investor landscape in gene therapy. Maybe just to share with you a few more statistics from the ARM first half 2020 report. There are now more than 1,000 companies working in the regenerative medicine space, exactly 1,001 as of June 30th. And of those, 515 are focused on gene therapy. So phenomenal growth in the number of companies that are operating in our sector. And also, just to give you a little bit of a sense, what the demand for clinical study financing and manufacturing facility financing, just in the first half of 2020 alone, more than $10.7 billion were invested in the regenerative medicine sector. And of that, about 50% of it was for gene therapy companies. And that's 120% more than what was invested in the first half of 2019. So I think, the companies that are operating in the space, both large companies, medium-sized companies, smaller companies, the rate of new company formation, the number of new programs that are being started, both preclinically and clinically, is continuing to expand. And, for me, now being in 6 years being responsible for our gene therapy business, I can tell you in that short period of time, and Ken, maybe you have a little bit longer window from when you started REGENX, and Kathy from when you've been both an academic and starting Spark. I mean, the rate of innovation and the pace of it is just unbelievable. I just welcome your thoughts on how you kind of view today versus where we were a few years ago, and what your expectations are for where we may be in the next 3, 5, 10 years from now.
Well, I might start just by saying that, I think this extraordinary explosion in the number of companies focused on gene therapy, I think may reflect the fact that when you develop the ability to either supply a gene or to edit a gene or knock down a gene or edit a base, then suddenly there's no undruggable target. Right? You can reach everything in the genome if you can devise a way to do it. And with the tools that are available, you can devise a way to do it. And so many things that we could not have thought about with other classes of therapeutics are now potentially within reach. And so I have to say that, from the time when we started out, particularly 20 years ago, when gene therapy was something that was pretty universally greeted with skepticism in many quarters, to see what's going on now is truly astounding. But I think that there's a firm scientific basis for that, as I indicated. I mean, there's so many things that you could do with gene therapy that we couldn't do with monoclonal antibodies or small molecule drugs or recombinant proteins or whatever.
I always love hearing Kathy's scientific approach to all these questions. It always comes back to the science for you, which I think is wonderful. For me, I start with the business case, and I think that there is a lot of money still going into biotech, even this year. And I think gene therapy is one of the darlings from an investor perspective. And there have been some really outstanding exits in the field, yours, mine, the Avexis, Novartis. And I think that there's an expectation that there are going to be more large pharma companies out there looking to place their bet in the field. And I think that that's what's driving a lot of the exuberance. I mean, 515 gene therapy companies, is that what you said, Bob?
Yes.
Yeah,
I was a little.
that's crazy. I mean, that's crazy. So I think, I mean, I tend to be a little bit Chicken Little. The sky will eventually fall and the spigot will turn off a bit. And I think there might be some winnowing of that, which I actually think will be good. I think evolutionary pressure, when there's money all over the place, you don't have to be better to get funded. You just say, I'm a gene therapy company. But if it gets a little tighter, it forces us all to be better. And so I'm astounded that this is where we are. I think it's a wonderful place to be. I guess I've been in the industry long enough to know that fortune's wheel kind of goes around and around, and at some point the music will stop a little bit, but I don't think that that will be a bad thing for our space, because it just means that it will force everybody to be that much more efficient, that much more innovative, that much more focused on value creation for patients, because that's what translates into value creation for shareholders. And so I think ultimately we're going to get to an even better place, even as the money dries up a little bit.
I probably land somewhere in between the science weighting and the business weighting, which is good for me to come in third here. I never thought about REGENX through the lens of, where am I going to get capital or how am I going to finance the company? And strong evidence for that fact is that I, started forming the company in August of 2008 in the worst recession in recent history of this country and maybe the world. By the time we decided to become a company that needed capital to develop drugs, there had already been other of our peers and partners, entering the process of financing. Ours was very accelerated, meaning we were kind of borrowing from some of the things that had gone on in 2013 and 2014. And, and then it changed everything. For me personally, I became much more aware of the process behind which, it propelled REGENXBIO into the public market and others of our peers. And I would say recently, I guess I found it remarkable because I thought it was a pretty energized time then, 5 years ago. It's as energized right now as it was, if not more so. When I made the transition from 15 into 16 and 17 and, got focused again on, okay, how do we build a business with science to treat patients for a very long time, which is a very different exercise than how am I going to take the company public. I started to communicate and reflect myself that consolidation has to occur, right? I mean, Natalie, to your point on the business side, I mean, even if the capital markets are strong, there's just no other place in our industry where you see in perpetuity sort of, single product companies, right? Just against the background of a platform. And, and Bob, your company and others, obviously through the reference to the acquisitions, by Astellas and by Roche. I mean, there has been an element of that, but I guess it is amazing to me that the reaction to that from a capital market perspective has been, well, let's seed more because, that consolidation is really not complete and needs to occur, on a smaller end of the spectrum. At this point, I'm a little bit concerned. I will say, it's exciting to see that level of support from the business side. But if I swing back to the pendulum on the science side, Kathy, are we over our skis a little bit here? I'm always looking over my shoulder and going, and I'm not certain, I think gene therapy is in a place and in an important place where it's beyond, one event, sort of really having us take even a full step back. I mean, there really is just too much out there on the platform from a safety perspective,
Sure.
From a clinically meaningful benefit perspective. But, as someone who's been feels really attached to this industry and gene therapy now for over a decade. You know, for patients, you know, I have a high bar for quality, and you know, you can't always be certain that the people that work on the financial transaction side have that same eye.
Well, you've certainly done a remarkable job, Ken, of building REGENX from its beginnings to the company that it is now. So congratulations on that. And we've now approached our time limit for the panel. So I would very much like to thank Kathy, Natalie, and Ken for joining us. And I hope all of the audience members enjoy the discussion and enjoy the rest of the meeting on the Mesa. Thank you.
Thanks, Bob.
Thanks, Bob. Thanks,
Thanks,
Kathy and Natalie.